Carboxylic acids can react with alcohols to form esters, a type of carboxylic acid derivative. This process is called esterification. Like always, we don't change how the carbons are connected to each other. The OH that's on the carboxylic acid gets swapped out for the OR of the alcohol. So in the example shown, the two green carbons of the ethanol molecule stay attached to the oxygen when it replaces the acid OH. Water is produced as a byproduct of this reaction. How do we know that this product is going to be an ester and not a hemiacetal or an acetal like we saw in chapter 15? It's always going to be based on the functional group. Carboxylic acids make esters, while aldehydes and ketones make hemiacetals and acetals. So you need to be able to identify the functional groups in order to understand the reactions. See if you can identify what each of the following products will look like before you continue. The top molecule has two carbons up to the carbonyl, just like in the carboxylic acid, but instead of the OH attached to the carbonyl is attached to the alcohol's oxygen and two carbons in that order. The second molecule has three carbons up to the carbonyl and then an oxygen attached to the isopropyl group, just like in the isopropyl alcohol that reacted with the carboxylic acid. Here's some more practice with esterification. Pause and see if you can draw out each product. I usually start out by drawing the carboxylic acid without the hydrogen, and then just add the alcohol carbon chain. So we have our 4-carbon branched carboxylic acid, but instead of hydrogen, we have a 4-carbon butyl chain, all in a straight line, just like in the alcohol. The second molecule reacts as a sec-butyl alcohol, so the ester ends up having that sec-butyl chain instead of hydrogen. It doesn't matter which side is the carboxylic acid, we still have to attach the alcohol carbon chain where the hydrogen was. This time the carboxylic acid is on the right side the way that we have it drawn. The alcohol carbons are isobutyl and they attach in the same arrangement to the oxygen instead of the hydrogen. This final molecule is actually the same as the top molecule, just drawn backwards. Esters follow a full set of IUPAC rules, just like everything else that we've talked about. But the easiest way to think about an ester's name is that it will come in two parts. The side that has the carbonyl, that is, the carbon-oxygen double bond, will always be named as if it were a carboxylate, and it will be used as the main chain name. In this case, it's the blue section on the left, propanoate. The carbons on the other side of the oxygen are listed as a side chain, as a separate word before the carboxylate. In this case, it's butyl, making the entire name butyl space propanoate. The first step from naming these molecules is to decide which side is the carboxylate. It's always the side with the double bond to an oxygen. For the molecule in the top left, the carbonyl is on the right side, so that's the main chain. It's 4 carbons making the main chain butanoate. Again, that's the part on the right. The 3 carbons on the other side are listed as a side chain, propyl, making the entire molecule propyl butanoate. If we have a side chain off of the carboxylate, we'll still include that as part of the name, just as we would with a normal carboxylate. The molecule on the top right has a carbonyl on the left side, so that's the side that gets the main chain name. We number from the carbonyl left, making a 3-carbon propanoate with a 2-methyl side chain, so just 2-methylpropanoate. We include that side chain as part of the entire main chain name for the carboxylate. The other side is butyl. See if you can name the last 2 molecules. The molecule on the bottom left is butyl 2-methylbutanoate. The butyl is on the left side and the 2-methylbutanoate is on the right. The final molecule is propyl benzoate. The carboxylate name, benzoate, is based on the name benzoic acid, one of the carboxylic acid names that we had to memorize. Just as esters can be formed from carboxylic acids and alcohols, they can be broken back down into those pieces by adding water. These reactions are slow without enzymes or acid or base to catalyze them. Look at the top reaction, which occurs in either water or acid, and compare it to the bottom reaction that occurs in base. What's different? Because carboxylic acids are acidic, if they form in base, they're quickly converted to the carboxylate. Otherwise, these reactions are identical, the top and bottom. Finding the product is easy if you can think about functional group conversions rather than trying to think about the direction of all the different carbons. The ester has two sides. One side has a double bond to oxygen, and the other does not. All the carbons up to the double bond to oxygen are going to be the same, but instead of being an ester, this chain of carbons becomes a carboxylic acid, or carboxylate. So the side with the double bond is going to keep a double bond to oxygen and be either a carboxylic acid or a carboxylate, depending on whether it's in water or acid or whether it's in base. The other side, without a carbonyl, will just always become an alcohol. All the carbons of that side of the molecule stay connected in the exact same way they were before, and the oxygen that was connected to those carbons becomes an OH instead of being part of an ester. So now for the two reactions that are shown below, pause and see if you can identify the two organic products, either carboxylic acid and carboxylate, and an alcohol. The carbons on the left of the top reaction are the carbons that become the carboxylic acid, because that's the side with the double bond to oxygen. The other side becomes an alcohol. We know it's going to be a carboxylic acid and not a carboxylate because it's in water, not a base like hydroxide. The bottom molecule is reacting with hydroxide, so we'd expect a carboxylate from the carbons on the left, which have the double bond to oxygen, and the three carbons to the right of the oxygen become a 3-carbon alcohol. Esters form from separate components frequently in biology. Small esters are often recognizable by their fruity odors, which, big surprise, are often found in fruit. Larger carboxylic acids, called fatty acids, esterify for both energy storage lipids and for membrane lipids. Some esters are also formed from an alcohol and an acid that are on the same molecule, a hydroxy acid. These cyclic esters are called lactones. The thromycin antibiotic drugs, all the ones that end in thromycin, are all some sort of lactone. You can see the ester in the lactone erythromycin highlighted in yellow on the structure shown. We talked about physical properties with carboxylic acids already. They were dominated by hydrogen bonding, but also had a very strong dipole, making them some of the highest boiling structures that we see. What do we expect to see change in an ester structure, like that on the right? Esters don't have an OH bond, which means they can't hydrogen bond. What do you expect that to do to the boiling point of esters compared with a similarly sized carboxylic acid? What do you expect that to do to the water solubility of an ester compared to a similarly sized carboxylic acid? There are tons of molecules that are very similar to esters with very similar properties. We see thioesters where the alcohol has been replaced by a thiol in molecules like acetyl-CoA and other similar molecules. These show up in the citric acid cycle in chapter 23 and in fatty acid metabolism in chapter 25. They function identically to normal esters. We also see phosphoric acid replacing carboxylic acids in esters. These often show up in protein and enzyme structures. Creating phosphoesters will drastically change the physical properties of that region of a protein because the phosphate is really acidic and really polar, and so will change the way that a protein will actually fold. We also see sometimes that phosphate esters are joined together. These are an efficient and stable way for our bodies to store up very short-term and very usable energy. When the esters are hydrolyzed, broke into pieces, the energy of that hydrolysis drives many processes in our bodies. The most common form these phosphates will take is adenosine triphosphate, or ATP, the most important form of cellular energy in our bodies.